Hello everyone, welcome back to my channel Bani's Biology. Today we are going to explore one of the most fundamental and fascinating topics in molecular biology, that is gene expression and gene regulation. So this concept explains how one genome can create thousands of different cell types and how organisms respond to their environment. If you have ever wondered how the same DNA inside every cell can create neuron, muscle cell and immune cell. So in this video, we will explore the mechanism behind the diversity. First, let's understand gene expression. So we all know that the process where the information stored in DNA is synthesized into mRNA is called transcription, and the process where the mRNA message is decoded to build a protein is called translation process. So together, transcription and translation convert genetic information into functional molecule. So this protein determine the structure and function of cell. So the process of making RNA and protein from a gene is called gene expression. It's called gene expression. So what is gene expression? So gene expression is the process by which the information stored in gene is used to produce a functional product, usually a protein and sometimes functional RNA like rRNA, tRNA and miRNA. So gene expression determine which protein a cell make and when they are made and how much is made. So gene expression is when a gene transcribed into mRNA and translated into protein. So this process is called gene expression. Here is an interesting part. All cells have the same DNA. That means all somatic cell have the same DNA. Like neuron, intestine, muscle, liver cell, skin cell all contain the same DNA. Yet they look different and perform different function. So here muscle cell, intestine cell and neuron cell, three type of cell contain the same DNA but they look different and they are perform also different because they express different set of gene. So they do not express all gene all the time. So this selective on and off pattern is when we call gene regulation. So gene regulation is the control of gene expression. So here muscle cell, intestine and neuron have different gene that are turned on and off different times and they express different set of gene. This is due to gene regulation. So here green light means gene is on, here red light means gene is off and here yellow light means conditionally active. So here different types of gene that express in different times. One more example, here is the skin cell nucleus, liver cell nucleus and brain cell nucleus. Here same gene is expressed but different level. Here gene transcribed at moderate level and here gene transcribed at high level and here gene transcribed at very low level. So different cell have different gene expression level and that determine the cell diversity, and this is called differential gene expression and it is the reason for cellular diversity. Gene expression itself can occur in different modes. Let's discuss different types of gene expression. One is constitutive gene expression, another is inducible and other is repressible gene expression. So in constitutive gene expression, where the gene is always on all the time under all conditions and these genes are expressed continuously. So these genes are expressed continuously because the cell cannot survive without their product, and these expressions do not depend on environmental signal because they perform basic essential housekeeping function such as energy production, basic metabolism, maintaining cell structure. Like an example, gene for glycolysis enzyme. So every cell types express these genes because every cell needs them. And also rRNA, tRNA and RNA polymerase subunit, those are essential for cell and every cell types express these genes because cell needs them. So the genes is expressed continuously. So this gene expression is called constitutive gene expression. Another is inducible gene expression where the normally gene is off but can be turned on when a specific signal called inducer is present, and these genes are activated only when the cell needs them. But in constitutive gene expression, the cell express these genes because cells always needs them because cell cannot survive without their product. But in inducible gene expression, when the cell needs them, then gene is expressed by the help of inducer. So here inducer help in the gene expression. Like an example, in E. coli cell, when the E. coli environment lactose is absent, then there is no need to synthesize the lactose digestive enzyme. So here structural gene lac Z, lac Y, lac A that express the lactose digestive enzyme. So in the absence of lactose, so there is no need to express lactose digestive enzyme. So here is gene expression is turned off. But when the lactose is present in environment, then lactose act as a inducer. Here lactose convert into allolactose and then allolactose bind with the repressor. In the absence of lactose, here gene expression is off by the repressor protein. Then repressor protein bind in the operator side and the transcription is off. But when the lactose is present, this time this allolactose act as an inducer and bind with the repressor protein, and repressor protein is inactivate, and this repressor protein cannot block the RNA polymerase moving, and then the lactose digestive enzyme gene is expressed, and the lactose digestive enzyme is expressed. When cell needs them, then the gene is expressed. So this is called inducible gene expression. So repressible gene expression when normally gene is turned on but can be turned off when the final product of the pathway becomes abundant. Like an example, in the absence of tryptophan, the cell keep the gene active because this product is needed for survival. So here tryptophan continuously express. That means the gene expression is on. But when the tryptophan is present, that like tryptophan is abundant, then this tryptophan act as a co-repressor. That means in the absence of tryptophan, the repressor is inactive. So this time the gene expression is continuously on, but when the tryptophan is abundant, so cells do not need them because the gene product already present. So here is the tryptophan act as a co-repressor and bind with the repressor protein. So then inactive repressor become active by the binding with tryptophan. So here tryptophan is the end product, and this end product act as a co-repressor and that activate the repressor and that bind with the operator side, and the gene regulation is off. That means gene regulation is off. So here first gene is turned on, gene regulation is turned on, and after the gene product becomes abundant, then the gene regulation turned off, and this abundant gene product act as a co-repressor. So here when the tryptophan is low, then repressor inactive and the gene regulation is on, and when the tryptophan is high, then tryptophan act as a co-repressor and bind with the repressor, and then this inactive repressor activate and bind the operator side, and the gene regulation is turned off. This is called repressible gene expression. So to understand how these expression patterns are controlled, we must first understand the types of gene involved in regulation. So there are different types of gene that are involved in regulation. First is regulator gene. So the regulator gene is a gene whose product control the expression of others gene. So the regulator gene is a repressor, activator, transcription factor, and this protein bind to the specific DNA sequence. This is the regulator gene, and this gene express the regulator protein that is activator and repressor, and this regulator protein that bind with the specific DNA sequence that is called operator site and decide whether another gene will be turned on or off. This is the regulator gene. And structural gene is the gene that code for functional product. This is the structural gene. This structural gene code for functional product, that is usually protein or functional RNA, and that perform a specific role in the cell. So here regulatory gene that control the other gene like structural gene, but here structural gene do not control the other gene. So structural gene is controlled by other gene. So this structural gene produce the protein needed for normal cellular operation like metabolism, transport, cell structure, enzyme activity, biosynthesis, and cellular function. So this is the structural genes. So another region of the DNA is promoter region. So promoter region is a DNA sequence where RNA polymerase bind to and initiate transcription. So promoter region also DNA sequence. So this is the promoter region. This is the promoter region where the RNA polymerase bind and initiate transcription, and it located upstream of the structural gene. This is the structural gene, and this is called upstream. So promoter region located upstream of the structural gene, and its function as the starting point of transcription and control binding efficiency of RNA polymerase. So another region is the operator region. Operator region is the regulatory DNA sequence where repressor protein bind and block transcription. This is also a DNA sequence, and this operator region present between promoter and structural gene. This is the operator region, and this denoted by O, that present between promoter and structural gene. Now that we know regulatory gene produce regulatory protein, activator and repressor. But what is activator and repressor? So this is regulatory protein, one is repressor, another is activator. So activator is a regulatory protein that turn a gene on, that promote transcription, and activator bind to a specific DNA site called regulatory protein binding site. This is the regulatory element or docking site where the regulatory protein is bind, that is repressor and activator. This is the repressor binding site, this is the activator binding site, and this binding site is called regulatory protein binding site. And repressor protein that turn a gene off. So the activator that turn a gene on, repressor that turn a gene off, and it blocks RNA polymerase and transcription cannot start, so genes stay off. So this is the activator and repressor. This is the regulatory protein. But this regulatory protein cannot operate independently. Its binding and activity are controlled by effector molecule. So regulatory protein binding depends on the presence or absence of the appropriate effector molecule. So what is effector molecule? Effector molecule usually is small molecules such as amino acid or sugar or similar metabolites, and that bind to regulatory protein and change its activity. So effector molecule act in two different ways. One is inducer, another is co-repressor. Co-repressor. Inducer is a small molecule that turn a gene on and it bind to the repressor and that inactivate the repressor, so that inactive the repressor and RNA polymerase can now transcribe the gene. So here is the repressor that transcribed from the regulatory gene, and this repressor first bind with the regulatory protein binding site and this time block transcription. But in addition of inducer, that is effector molecule, this inducer bind with the repressor and this repressor become inactive, and this repressor cannot bind the regulatory protein binding site, and then the transcription is on, and this system is called inducible system. Inducible system. Next is the co-repressor. Here is the regulatory gene that synthesizes the regulatory protein, but this repressor cannot bind to regulatory protein binding site because this repressor is inactive. So first the gene regulation is on. After addition of co-repressor, the co-repressor bind with the repressor, then the repressor become active, and the repressor bind with the repressor binding site and that block the transcription process. So first the gene is on, after binding of co-repressor, then gene becomes turned off, and this system is called repressible system. So this is called inducible system and this is called repressible system. So here regulatory protein is controlled by the effector molecule, that is inducer and co-repressor, and this system, one is inducible system and another is repressible system. So inducible system and repressible system ultimately function through two major regulatory approaches. One is positive control and another is negative control of gene expression. So in the negative control of gene expression, in negative regulation illustrate for both inducible system and repressible system. So in negative inducible control system, where the repressor bind to the regulatory protein binding site and prevent the transcription of the structural gene, that means block the transcription in the absence of inducer. But in the addition of inducer, the inducer bind with the repressor and the repressor become inactive. So repressor become inactive and that cannot bind to the regulatory protein binding site. Then the transcription is on. This is the negative inducible system. But in negative repressible system, where the repressor cannot bind to the regulatory protein binding site because the repressor is inactive. But after addition of co-repressor, the repressor become active and that bind to the regulatory protein binding site. So when the repressor is inactive, the gene regulation is turned on, but when the repressor is active, then the gene regulation is turned off. So in negative control of gene expression, where the repressor protein is used to regulate transcription. Repressor protein is used to regulate transcription. But in positive control of gene expression, where the activator protein is used to regulate transcription. Here also present inducible system and repressible system present. So here in inducible positive control of gene expression, where the activator is used. So in inducible system, where activator is inactive, but the RNA polymerase cannot bind to the promoter without the activator at regulatory protein binding site. But after addition of inducer, the regulatory protein become active and then regulatory bind with the regulatory protein binding site and the RNA polymerase is bind and then the transcription is on. So here activator protein that help in the activation of gene expression. When the activator is inactive, then the gene expression is off. When the activator is active, then the gene expression is on. That's why it is called positive control of gene expression and it is called inducible system because the inducer that help in the activation of activator. But in repressible system, where the regulatory gene that synthesize a activator protein, but this activator is active, that bind to the regulatory protein binding site and the gene is on, then the transcription is on. But after addition of co-repressor, then co-repressor bind to the activator and the activator become inactive. So if the activator is inactive, then the RNA polymerase cannot bind with the RNA polymerase binding site and the transcription is turned off. So here co-repressor act as a repressor. So this is called repressible system and this is called inducible system, and this is called positive inducible gene expression. This is called positive repressible gene expression. So here in positive control of gene expression, activator is used. So in inducible, activator first is inactive. After addition of inducer, the activator become active. But in repressible system, first activator is active, but after addition of co-repressor, the activator become inactive. So that's why I call repressible system, but it's positive control of gene expression. But in negative control of gene expression, where the repressor protein is used. So here in repressor protein, where the inducible system, first repressor that bind to the regulatory protein binding site and transcription is off. After addition of inducer, then transcription is on. That is called inducible system. And negative control of repressible system, where the regulatory gene that synthesize regulatory protein that is inactive. Then the transcription is on. That cannot bind to the regulatory protein binding site. And after addition of co-repressor, that bind to the repressor and become activator, and that bind the regulatory protein binding site, then the transcription is off. So this is the negative control of gene expression, where the repressor is used, and in positive control of gene expression, where activator protein is used. So at its core, gene regulation is about balance, turning gene on when needed and silencing them when not. This balance is what allows a single genome to create the diversity of life we see in tissue, organs and entire organism. And understanding this balance brings clarity to the complexity of biology. If you found this video helpful, make sure to like, share and subscribe for more clear and high quality biology lessons.